Hey everyone, so today we're going to talk about lipids and nucleic acids. I always find these videos really funny to begin with, like so formal to introduce yourself and until you find your flow as a teacher again, no matter how many times they do them. Today I've got my little furry friend here. I just bought a little mic. I was told that the audio would be better from this than it is from my MacBook. So, give me a thumbs up if you found that was something that made an impact for you. Um, if not, I have to try a different mic, I suppose. But I've seen lots of people, these furry friends, holding them close to their mouth. It's very sensory, actually, to touch. Probably made some crackling noises there. But cracking on with today's lesson. Now, this is a free resource while I'm talking through here. You'll find the link below in the description. There's also a link to a multiple choice test for you to test your knowledge at the end, all based on an AP style, so assessing the same cognitive strands of application. Don't forget that I've also got my free site, cramney.study. For those of you new to the channel, just check it out. I really love the AP. Oh, I forgot to write HTTPS at the beginning. HTTPS. It's got a really nice set of cards for AP with application as well as, so here we go. So the way I've set it up is some definitions and application. So like we're looking at unit 1 here. You could browse them. You can star cards that you want to come back to up in this function here. So why does sweating actually cool you down? Then you can have a look at the answer and self-assess here. I use the same approach. It's called space repetition. I use the same approach for learning French. And I can say I'm definitely moving in the right direction. Like I know it works. It's based on science. And cracking on, we'll start with lipids. So it's quite a short set of information to learn today. There's a few facts about lipids that's really important, and we'll talk about how that is assessed as well. Now, lipids, the key thing to remember is they are non-polar. That means that they are hydrophobic. So that's why I called them the molecules water will not touch. So if you add like olive oil or vegetable oil to water, you'll see that it forms a layer on top. It's less dense, so it floats, and it's also hydrophobic, so it repels water. Now, this is a fact here. So we've got no true monomers when it comes to lipids. Remember cho, chon, chomp. We also spoke about the difference between a monomer and a polymer. So monomer is a singular unit, and a polymer is where it's built up of lots of individual ones. So there's no true polymers in lipids. And if we look at the CHO, so it's CHO for carbohydrates, CHO for lipids, but there's the silent P that we put in brackets, and that's because of phospholipids. So phospholipid would be this guy down here. So phospholipid is what makes up a phospholipid bilayer. So here's our phospholipid bilayer. You can probably spot the mistake here with this one straight away. There should be 2 fatty acid chains on each one of these. I'll fix that so when you view the video, it's not incorrect. But it's correct down here. Wait, no, it's not. I swear to God it was earlier. It'll be correct when you actually view the video. I can't believe it. But like every phospholipid has this glycerol phosphate head and then 2 fatty acid tails. And these are ester bonds holding them together. Now, what's important with a fatty acid is it straight or is it got a kink or bent? So when it's straight like this, it's fully saturated. That means that each one of these little points here, at the angles, these would be carbons, and then each carbon can form 4 bonds. So it's attached to 4 hydrogens. It's fully saturated. Sorry, not attached to 4 hydrogens. Carbon either side and 2 hydrogens. So it's fully saturated with bonds. And if it's unsaturated, so here this is representing a double bond. This double bond here means that there's a kink. And when it's kinked like that, it's because it's what we call a cis unsaturated. So cis, I always put my double bond as my 2 thumbs, and then these 2 sticking up here are my hydrogens. So a cis is where the hydrogens are on the same side, and that forms a kink. And if it's trans, it's like this. So the double bond in the middle, and then 1 hydrogen's down, 1 hydrogen's up. Then that is actually straight. So trans is something that we have actually artificially made. It's called, trans fats. You've probably heard of them. They're banned in some countries because they are found to be. I'll try and remember to edit that bit out as my dishwasher going. So trans are straight. And then that means that because of if things are straight and they're not kinked, they can be packed tighter together. So you can see them like sardined in there. There's more London dispersion forces, so that weak intermolecular force of attraction between the fatty acid chains, and therefore they're found as a solid at room temperature. And the same for trans, so similar to saturated. Um, Whereas an unsaturated cis, or like polyunsaturated ones that are cis, this provides a kink, and that kink adds fluidity, which means they're then liquid at room temperature. Some other things to note is that yes, we don't get polymers. Glycerol plus free fatty acids joined by ester bonds in a condensation reaction that releases three waters assembled but not a repeating chain. So they're not true polymers. And then we have twice the energy packed in to what you'd have with a carbohydrate, and they are stored dry. And then we want to think about fluidity. So how this could be assessed would be thinking about lipids as a membrane, and we'll come back to this when we talk about cells a lot more, and how temperature affects the fluidity of a membrane and how the kinking affects it. So this is saturated. Remember, there should be two tails each one. There will be in your interactive one you can look at. I'm fixing the bug at the moment. And you can see there's some slight movement because all molecules are moving. Now, if it's unsaturated, then there is a kinking like this, and that forces space between the heads. That means it's more fluid. Now, things that can move through the membrane are small non-polar molecules like gases. And this is a whole unit in itself. So oxygen, carbon dioxide is now able to pass easier through this. Other big things that can pass through slowly will also be passing through easier now. If it's cold, everything gets more packed together and there is less movement. So how do we create space when it's cold? We can actually add another amphipathic molecule, because these molecules that have a like hydrophilic head and a hydrophobic tail that have this dual charge here, we call them amphipathic. And then another one would be cholesterol. So cholesterol is something that's got a hydrophilic head and a hydrophobic tail, and it packs into the membrane. And when it's cold, it's going to increase the space between the phospholipids and make it more fluid. And when it's warm, it actually works the opposite way, and it pulls things together that are spreading out too much, and it increases the attraction between them. So we say that it buffers or modulates the fluidity of the membrane because it works differently depending on the temperature. You might be asked about this in the context of, like, I've seen questions about the rate of diffusion of gases when it's been cold, and what might you find in the membrane of a fish that lives in like cold water. So if it needs to increase the fluidity, there's two things. You're going to increase the amount of cholesterol to cause the spreading out of those phospholipids, and there's going to be more unsaturated fatty acids to increase the kinking, and therefore increasing the space between the molecules. So moving on now to think about nucleotides or nucleic acids. Nucleotides are the monomers of nucleic acids. So we had chon, and chomp right at the end, CHONP, carbon, hydrogen, oxygen, nitrate, and phosphate. That's our nucleic acids. So the building blocks of those contain 5 different elements. And they're found in each one of the nucleotides or each one of the monomers. And we've got 2 types of monomers. We've got a DNA monomer that has a deoxyribose, and we've got an RNA monomer or nucleotide, which has a ribose sugar. So you can see here the second carbon, second carbon here, we have hydrogen at carbon 2, and there's an OH group at carbon 2 when it's a ribose. So this is lost an oxygen, deoxyribose. Now, it's really important to number our carbons, 1, 2, 3, 4, 5, because when we talk about nucleotides and then we talk about transcription, translation, protein synthesis, we're thinking about the movement of enzymes in certain directions from like a 5 to 3 end, or some things happen in a 3 to 5 end. Now, here you can see the phosphodiester bonds runs from the carbon 5 to 3. So a strand has a head and a tail. Sequences are always written 5 to 3. That means that, like when we say something is synthesized, it's actually adding to the 3 end. When we say 5 to 3, like if we go A, T, G, C, so say this one's G, if we add another nucleotide, we're adding it to the 3 end. So that's a 5 to 3. The enzyme moves and 5 to 3 and adding this side. But it's having to read whatever is on the other side. So it's reading 3 to 5. That gets a little bit confusing, and we'll do a whole video on that in itself. But for now, it's just really important to know that we number our carbons. That helps us to describe the direction of movement for enzymes, the direction that enzymes work and in which they read, and also the formation of bonds. Like when we talked about glucose to glucose, is a 1,4 glycosidic bond, or is it a 1,6 glycosidic bond? And how does that affect the growth structure of the polysaccharide? So the other thing to note with nucleic acids is when we talk about DNA, we have the bases T, A, C, and G. And when we talk about ribose or the RNA, we have U instead of T. So we have U, A, C, and G. Then we have some base pairing rules and the directionality. So here is our molecules of DNA. If we have a look here, what you might notice is that you get a different number of bonds between C and G. We have 3 hydrogen bonds, and between T and A, we have 2 hydrogen bonds. This gets examined in a way where they're like, here's 2 samples of DNA. Which sample separates easiest? And if there's more bonds, it separates harder than if there was less bonds. It would take less energy to break those bonds because there's less of them. So if we've got a sample heavy in G and C, then it's going to take a higher temperature and more energy to break those 2 DNA strands apart. Directionality is something we come on to more when we talk about protein synthesis. But you can see here, this is our 5 to 3 end. This is our 5 to 3 end in the opposite direction. So with DNA, we say that this has this directionality to it, and it's moving in anti-parallel. So they are parallel, but one is inverse, so it's anti-parallel. Nice little mind map for you to have a look at how it all fits together down at the bottom. One thing that I'll point out at this junction here is this pink line here. So water excludes both. And that's just meaning, when we have this non-polar town DNA, they are both hydrophobic. And you can have a go at filling in the gaps. And we've got some sample AP style questions here that are different to the other ones in the practice MCQ test that I'll share at the bottom. I'll be building out FRQ ones at a bit of a later date, but at the moment I'm moving in many directions. And I think MCQ is something that are really nice to start with at the beginning of the course. FRQ with AP is something I focus on later on once we have more knowledge that we can pull together, because ultimately the way that they test it is multiple different units kind of interwoven together. So it's hard to do a lot of AP FRQ practice right now. So a sample of DNA is found to contain 30% adenine. What percentage of the sample is guanine? So if adenine pairs with thymine, so 30 plus 30 is 60. And then G pairs with C, so divide 40 by 2, we've got 20%. And here's what I was talking about with different strands. So 2 DNA samples at equal length are heated until the strands separate. Sample X separates at 78 degrees and sample Y at 92 degrees. What is the best explanation of this? So if it took more energy to separate the strands, there's probably more of those triple bonds, and there's more of those triple bonds of hydrogen bonds in the G and C. Okay, so remember to subscribe. I'm going to leave it there. Yeah, so First of all, I will leave it here for today. I'll let you finish the rest of these questions on your own. And remember to like and subscribe and share with your friends. And of course, check out my free app Cramly. This will never be token gated. This will never be, yeah, like you'll never have to pay for this. So at this point in time, and forevermore, this will be free to you the whole year. And hopefully it's something that's really helpful. There's some fun games in here too. So like Helix, you got a daily Wordle style problem, quick 5-letter word. Let's think ribose. Oh no, it's not even 5 letters. Helix is boom. Even got the nice animation. So yeah, hopefully you find this helpful, and tune in for the next one, which will be on protein. So ciao for now, and have a good day.